To everyone, and thank you for your support of our third out of 4 talks for the Metabolomics mini lecture series. This morning, we have Assistant Professor John Paul Kovalik to talk to us about using targeted metabolomics to study human diseases. Dr. Kovalik received training from Duke University at Durham, USA, and he is serving currently as the director of the Duke-NUS Metabolomics facility. He is also an associate consultant at the Singapore General Hospital. And most importantly, he's our resident clinical and biological consultant at our facility. You are most welcome to visit him to get your advice on clinical, experimental design or metabolomic study free of charge and no minimum spending required at our facility. Over to you, JP. Thank you.
Okay, thanks, Chenhong. Thanks everyone for spending time with me. I hope it'll prove worth it. The title today is Understanding Targeted Metabolomics and its Use in the Study of Human Disease. Let's see if this works. Okay, I got to the next slide successfully, right? So I'll talk about some metabolism pathways and then how we can use targeted metabolomics assays looking at these pathways to help understand human disease better. And when I conceived of doing this talk, I thought about the conversations I have with people who come to our facility, who use our assays and then get some results. And then they come to us to talk to us about the results and try to understand and interpret the biology behind that. So I'll try to talk more about the biology underlying these metabolism pathways and how they might be contributing to disease. Issues such as data analysis and actually study design, I'll leave to other people. In particular, JJ, my first plug for next week, one of our colleagues, JJ, is going to talk about data analysis next week. So first, again, a quick review of targeted versus untargeted metabolomics. What we're talking about today is targeted metabolomics approach. And the take-home message here is when you do a targeted approach, that means that before you start, you decide what you're going to measure. So you say, these are the things that I want to measure, and I'm going to set up my assay to measure these things very well. Then I go ahead and measure them, okay? So there are some advantages to this. You can pick and choose the things you want to measure and make sure you measure those things well if you think they're important. With this, you can do things like absolute quantitation. So you can know exactly how many micromoles or nanomoles or picomoles there are of that metabolite in your biologic sample. This, I think, is especially important when you do these large human clinical cohorts where comparing between study groups or trying to set up some kind of threshold for diagnosis, you need to know an absolute number. A couple other things that make this more of an advantage is that you can pick and choose low abundance metabolites, so things that are very low concentration that can be missed by other methods. You can design your assay to specifically look at things that you know are not around in very high amounts, but are important. And again, because of use absolute quantitation, I think they're pretty suitable for large clinical cohorts. And then in contrast, there's untargeted metabolomics. What's nice about untargeted metabolomics is it takes what is considered an unbiased view. So you look at everything in your sample, okay? So this is very good, especially if you want to have new target discovery, right? A drug or a chemical which has not been described before or not been associated with a disease can pop up on your untargeted metabolomics platform. There are some issues with it. First of all, it's not fully unbiased because the way that you prepare the sample and the way you run it on your machines defines the things that you can measure successfully. So it's very difficult to measure all the different classes of chemicals in one sweeping assay. So you have to take that into consideration. You don't get absolute quantitation, so you get relative abundance when you compare experimental versus control. So you have to be very careful when you set up your experiment. What are your controls and so forth. And then it tends to have a bias towards high abundance compounds. So the key is, if you can see my pointer here, when you do untargeted, you get a bunch of peaks in your control sample, and you get a bunch of peaks in your experimental. And if you have a teeny tiny peak here, which is different from one or the other, it can be very difficult to discern those tiny peaks, those low abundance differences in low abundance compounds. So those are some of the drawbacks. What I'm going to talk about today are some targeted metabolomics panels that we do in our lab. So these are panels we set up where we predefine what we're going to measure, and then we measure them for you. Okay, so last week, I think Jan Hong showed you this figure. It's a table of a figure of most of the important central carbon metabolism pathways in human tissues. And so one thing we do with targeted metabolomics is we very specifically look at important pathways and make sure that our panels can look closely at some different important pathways. To try to simplify it, I've redrawn it, taken out a few chemical intermediates to show you the things that we look at. So we mostly concern ourselves with fuel metabolism, so things, carbon molecules that fuel tissues and cells, and they are kind of demonstrated here. So one thing we do is we can measure glycolysis. I'm not going to, and the pentose phosphate pathway. I'm not going to talk too much about glycolysis and the pentose phosphate pathway because in interest of time, I don't want to run over. And we haven't used this very much, especially in human clinical cohorts yet. So I don't have good examples of where this has been applied successfully in these human clinical studies. I will talk about the amino acids, fatty acid metabolism pathways that bleed into the organic acids, which center around the mitochondria. Okay, so now I'm going to go into these different classes of compounds. The first one are the amino acids. And again, our assays look at amino acids, and then we also, because of the other things that we look at how amino acids feed into catabolic pathways. So, you know, there's a bunch of different amino acids in the body, and they can all be broken down as fuel. And this is a graph demonstrating how that can happen. Now, there's a special class of amino acids, which I have to talk about here because it figures important in physiology and also when you do these human clinical cohort studies. And these are the branched chain amino acids. They're somewhat unique in that their functional group, the R side chain, has a branch carbon chain in it, and that makes their metabolism a little bit different. And so, in fact, the branched chain amino acids have a very unique pathway for catabolism in the body, which separates them out from the rest of the amino acids. And that's demonstrated here on the right. So this is a diagram showing what happens when you ingest protein and absorb it into the body. So the protein meal gets into the gut. It's broken down into eventually individual amino acids, which enter the portal circulation to go to the liver. So this is supposed to be a liver here. Now, all the amino acids can be broken down in the liver to be burned as carbon fuel or reformed as protein or what have you. All of them except for the branched chain amino acids. So it turns out that when you break down any amino acid, one of the first steps is often a transamination step. And the enzyme that does the transamination, does the initial breaking down of the branched chain amino acid, it's called BCAT, branched chain aminotransferase. This enzyme is not expressed in the liver. So the liver cannot process the branched chain amino acids. They pass right through the liver into the general circulation. So this is unique for the branched chain amino acids. All of the amino acids enter the liver and can be broken down and used, but not the branched chain amino acids because the liver lacks this enzyme. Now, the tissues that do have this enzyme are skeletal muscle and, interestingly enough, adipose tissue. Although no one talks about adipose tissue BCAT very much, the focus seems to be on the muscle. So we'll talk about that, I guess. So these branched chain amino acids bypass the liver and go to the muscle. And the feeling for the branched chain amino acids is they're sort of a nitrogen source, a nitrogen store that is kept and kept in the muscle for future use. So the muscle is a place where your body can store nitrogen and amino acids for when needed in a pinch. And the primary source, the primary input of this nitrogen is via the branched chain amino acids. So they get into the skeletal muscle, they get deposited within the skeletal muscle tissue. And when there's a need for the body to use amino acids or nitrogen, muscle tissue gets broken down, you release these branched chain amino acids. They either directly enter the circulation or the nitrogen gets transaminated, moved into glutamine and alanine, which then can circulate throughout the body. So the branched chain amino acids are unique in several ways. They bypass the liver to be stored in the muscle, and they're sort of the body's nitrogen repository in the skeletal muscle to be mined when needed. Okay, so why did I make such a big deal about branched chain amino acid? Well, it turns out it's pretty important for metabolic disease. This is from a paper that is now over 10 years old. And this was done at Duke with Chris Newgart's group, and they had a large cohort of people who are either lean or obese and insulin resistant, and they did a targeted metabolomics panel where they're looking at amino acids. And what they found was that in the subjects who are obese here, the branched chain amino acids valine, leucine, and isoleucine were higher, so increased concentrations compared to the lean insulin sensitive folks, okay? Now, this is actually not the first time this was observed. This was reported by Phil Felig, who was at Yale in the 1970s, was when he made this report. And Chris Newgard brought this back up. And it turns out that this signal of elevated branched chain amino acids in the circulation in insulin-resistant people is a very robust signal. Most studies that look at amino acid levels in the blood of people with insulin resistance will show this elevated level of the branched chain amino acids. Now, what exactly does that mean? Well, I mean, people have been looking at this for a long time. It's not quite clear. So here is a review article showing this diagram of what happens to BCAA, where they go, what might be doing. So not quite clear, but the signal is definitely there. So this amino acid, which is physiologically probably a way for the body to store nitrogen in the skeletal muscle and released upon need, is elevated in people with insulin resistance. The other thing I will talk about with amino acids and nitrogen is the story of choline and TMAO. If you had listened to the talk done by Jan Hong and In Swan, I guess last week or 2 weeks ago, she went into great detail into the story. So it was a very interesting story. It was done by this group at Cleveland Clinic. They did an untargeted metabolic metabolomics assay in patients at high risk of cardiovascular disease. And what they found was elevated level of this compound called TMAO, trimethylamine oxide, in the blood of people with high risk of cardiovascular disease. So this is kind of interesting because this is a nitrogen-containing compound, and it's derived from, what they showed in their studies is derived from the gut, from taking in a food that is high in nitrogen-containing compounds. So here they show red meat and eggs, but typically red meat is very high in these nitrogen-containing compounds. They get into the gut, and what they show is that your gut bacteria can act upon these nitrogen-containing compounds and make trimethylamine, which is a methylated amine, goes into your body and acted upon them by the liver to make this TMAO. And then they further showed in experimental mice that this TMAO stuff is pretty toxic to the endothelium, especially around the heart. So it does something to macrophages in the endothelial wall, especially around the heart, and causes buildup of plaque and probably cardiovascular disease. So this is an ongoing story. It's a pretty interesting story. We'll see where it leads to. It's also interesting because it provides, it provides a link with this well-known association between high intake of red meat and increased cardiovascular disease. So I'm getting older, but even when I was young, I remember these health ads on TV saying, reduce your amount of red meat because it's going to cause heart attacks. And people always thought because red meat has cholesterol, and that's why. But here's a different story, perhaps why increased consumption of red meat is linked to cardiovascular disease. It's from the high abundance of these nitrogen-containing compounds, which your gut bacteria and your liver can turn into this TMAO compound, which is not so good for your heart, apparently. I think this is not the full story about nitrogen compounds and human disease, especially cardiovascular disease. I think there's more to it than just TMAO. And in fact, within our lab, we're now in the process of developing a targeted metabolomics assay to look at all the different nitrogen pathways in the body. And we should have that nitrogen panel finished sometime next year. I'm looking at you, Jan Hong. I'm interested to see what we can do with this panel, especially when we look at human clinical cohorts and see if there's something else going on with nitrogen pathways and human disease, particularly human cardiovascular disease. So we're okay, we can keep on going, Jan Hong? Okay, so now I'll talk about the organic acids. So the organic acids are one of the metabolites found inside the body. They're generally associated with the TCA cycle. So most of the organic acids that we measure in our lab are associated with the TCA cycle, as well as a few other organic acids. And these organic acids are important because different fuel sources like amino acids, purines, odd chain fatty acids can feed into the TCA cycle at various points. via these different organic acids to help fuel the mitochondria, okay? I'm going to give a few examples of organic acids and their involvement in disease. The first one is really something done by the basic scientists, and it was based on these observations that a lot of tumors that they find, especially ones that have bad prognoses, have mutations in this TCA cycle enzyme called isocitrate dehydrogenase. So isocitrate dehydrogenase, or IDH, normally converts isocitrate to alpha-ketoglutarate. That's the normal function. And that's shown here on the right. My pointer is trying to highlight that pathway. So lots of tumors have mutations in IDH, and it seems to portend a bad prognosis. And originally it was thought, well, it doesn't work properly. But it turns out that these cancerous mutations in IDH1 work just fine. What they do is they do a different type of enzymatic activity. So these mutated cancerous IDH mutations, instead of taking isocitrate and converting it into alpha-ketoglutarate, these mutated enzymes take alpha-ketoglutarate and instead convert it into something called 2-hydroxyglutarate, okay? So this is a new enzyme activity that is encoded in this mutation in this enzyme. So this has been termed a neomorphic activity. So the cancer-associated mutation causes the enzyme to have a new activity, a neomorphic activity. And the new metabolite that it makes is now being called an oncometabolite because it's only produced in these cells that have this particular enzyme mutation. And you can see here on the left, this is sort of a mass spec elution profile. And this is shown on the top here for these AML cells that have with a wild type IDH. And the bottom, AML cells that have the IDH mutation. So these bottom cells are kind of nastier, right? A nastier form of cancer. And you can see here on the top, you see an elution profile where you see aspartate and glutamate. And suddenly on the bottom one, in this mutated form of cancer, you see the sudden appearance of this 2-hydroxyglutarate. And then there's been a lot of biology to try to understand how this oncometabolite 2-hydroxyglutarate is making things worse. And there's lots of ideas about interrupting the hypoxia pathway or altering protein, DNA methylation pathways. I won't go into it because it's actually getting quite complicated. So that's organic acids in cancers. There's other examples of organic acids in cancers, particularly this well-known phenomenon of the Warburg effect. So in lots of cancers, despite the fact that oxygen levels are lower, the cancers seem to stop using the TCA cycle and produce lots of lactate instead, called the Warburg effect. And people are still trying to understand why this is important for certain kinds of cancer growth. So moving on from cancer, but sticking with organic acids, I'll talk about the ketones. So the ketone bodies are an organic acid sort of, and we measure it as part of our organic acid panel. Ketones are becoming interesting for several reasons. One here I'm showing is the title from a paper from 2016. Now this is kind of interesting. This has to do with SGLT2 inhibitors. SGLT2 inhibitors are a drug used to treat diabetes. They're kind of funny how they work. What they do is they make you pee sugar in your urine. So you lower your blood glucose by peeing it out of your kidney into your urine. And when this first came out, I thought, well, I don't know. I mean, I guess it lowers your blood sugar a little bit, but big deal. We got better blood glucose lowering drugs out there for diabetes. But then they went and they looked closely at cardiovascular side effects of this drug. Nowadays, whenever you have a new diabetes drug, you have to prove that it doesn't cause cardiovascular deaths. There's a long and interesting story related to this. Now, to everyone's great surprise, when you give SGLT2 inhibitor drugs to people with diabetes, you markedly reduce cardiovascular events and markedly reduce worsening of diabetic kidney disease in these patients. So these SGLT2 emitters, which cause you to pee out sugar, somehow protect your heart against, I don't know, heart failure, I guess, and protect your kidney against diabetic kidney damage. And no one understands how this works, okay? So this first came out maybe 5-6 years ago, this data, and people were scratching their heads trying to figure it out. One well-known effect of the SGLT2 inhibitors is that it increases the level of ketones in your blood. So SGLT2 inhibitor causes a mild ketosis. And then one of the side effects is it causes a not so mild ketosis, which can make you very sick. But some people have hypothesized that the increased presence of ketones in the body is good for you, and that's what helps protect your heart and your kidneys from the damage of diabetes when you take this drug. And this is a paper that was done in 2016. Basically, it's a paper in diabetes care where the author just hypothesized that ketones are good for you. And here is sort of a diagram. He shows that when you give SGLT2 inhibitors, it changes fuel metabolism in the heart, and it makes the heart use a lot more ketones for heart work, and that this is beneficial for the heart. So this is an example where the measurement of an organic acid has potential importance for the study of human disease. In this case, it's diabetes, and the continuing conundrum of how SGLT2 inhibitors protect people with diabetes from heart and kidney problems. This is one of the, I think, the great unsolved questions in diabetes right now. How do SGLT2 inhibitors work, and how do they, we know how they work, but how do they protect, sorry, I was pushing a button, how they protect the tissues from diabetes damage. Ketones are also of interest to cardiologists. I see more and more cardiologists talking about ketones as a metabolic fuel and how it might be helpful, especially in the case of a failing heart. Now, just before I belabor organic acids too much, I just want to show you this picture. Some of you may be clinicians or clinical coordinators. You would recognize this as a 24-hour urine jug. So we give this to patients and we tell them to pee in it all day long for a whole day. So we collect all your urine for 24 hours. And we do this for a reason. It's because your body makes all these things that appear in the blood, and a lot of it gets filtered into the urine. So the urine is a good place where the biochemistry of the body gets captured. And if you collect all the urine for 24 hours, you get a nice integration of what's happening with your body's metabolism over a 24-hour period. So whenever you have disturbances in enzymes involved in fuel metabolism, you can alter the appearance of different metabolic intermediates. You can think of where a pathway is disturbed. Upstream of that enzyme, you see accumulation of the compounds, and downstream, you see less of it. And so all these metabolic intermediate pathways can get disturbed. And a classic place where we see this is in the class of compounds called the organic acids, which are a big part of central metabolism. And so when you have perturbations in central carbon enzymatic pathways, you will see build up or drop off in the level of the organic acids. And one place where you can see it quite easily is in the urine. And in fact, initially, these humans who have enzyme defects in central carbon metabolism pathways, these were diagnosed by taking urine and measuring the organic acids in the urine. And so these inborn errors of metabolism, they're called, are also sometimes called organic acidurias, because one of the prominent findings is dramatic changes in the level of organic acids in the urine. So here on the right is sort of a overview of some of the different diseases. I don't remember most of these disease names, but typically what you see is worsening function, especially in newborn babies. Babies are born, they appear healthy, and then suddenly they start to get sick. And many times this is because these babies have some new mutation in the enzyme of metabolism. Now this, by the way, is the reason why when your baby is born, the doctors take a heel prick of blood and they send it off to a central lab. So I think they do this in Singapore, they do this in the US and most Western countries. All newborn babies they take a blood sample from all newborn babies and they screen for the presence of these types of metabolic disorders, because sometimes if you catch them before they get sick, you can do something to prevent worsening disease. Now, these organic acidurias are important for people who have very dramatic perturbations in these metabolic pathways. But it's possible that you can also see changes in urine organic acids when you have mild changes that don't make you very, very sick, like show up, near death in the hospital, but perhaps contribute to disease. So this is a table from a paper done by our good friends and colleagues from Khoo Teck Puat Hospital here in Singapore. The study was headed by JJ Liu, who will be talking next week, and Lim Soo Chi, where they were looking at a cohort of patients with diabetic kidney disease, and they measured the urine organic acids and found that there are changes in the urine organic acids, particularly in the levels of fumarate, which can predict which patients have more severe progression of their diabetic kidney disease compared to other patients who don't have such marked changes in their organic acids. So the take-home message here is that the urine is the great place that collects and integrates all the changes in metabolism in the body. And screening the urine could be a good place to start off when you want to look for potential mild or hidden changes in metabolic pathways that might have importance for your disease. Okay, so this is sort of the meat of the talk, so to speak, the acylcarnitines. I'm going to talk a lot more about acylcarnitines because it's probably the one assay which most of our users use most. And our experience has been, especially in large human clinical cohorts, if you're going to see a change in metabolism pathway in your disease subjects, the most likely place to see it is in the acylcarnitines. So for whatever reason, the acylcarnitines are very sensitive to changes in human physiology. So if you have some kind of disease state where there's a change in function or some other change which is detectable, you can very often see it in the acylcarnitines as well. So it's a great place if you have a large set of blood samples from healthy controls and diseased people. It's a great place to start looking is in the acylcarnitines, because our experience has been that you very frequently find signals here that you wouldn't find elsewhere. Now, the acylcarnitines are sort of a way to look at fatty acid metabolism. So fatty acids generally are released from the adipose tissue as free fatty acids. Fatty acids are also released from the liver in the form of VLDL triglycerides. You can also get fatty acids from the chylomicrons in the gut, and they circulate in the blood, are taken up by the tissues. Here it shows skeletal muscle. Now, in terms of the tissues that use fatty acids and generate acylcarnitines, the big two users in humans are skeletal muscle and liver. So when you have looking at the acylcarnitine profile in the serum of human subjects, what's conditioning that blood, the two main tissues that are inputting the most signal are the liver and the skeletal muscle. So something to consider. What's interesting is that even in human diseases where liver and skeletal muscle are not the primary site of action, so the primary site where the disease manifests, we still frequently see perturbations in the acylcarnitines, even though liver and skeletal muscle are what are mostly generating the acylcarnitines you see in the blood. It could be like an earlobe disease, right? It only affects the earlobe, but there are cases where we have this kind of isolated tissue disease where we still see changes in the acylcarnitines, suggesting that there's probably broad, system-wide changes in metabolism that manifest in the ear, but are also affecting these other tissues enough to see a signal. Okay, so fatty acids are burned as a metabolic fuel in tissues, okay? And it's sort of kind of interesting, but human physiology, mammalian physiology, they like evenness. So when you burn fatty acids by removing 2 carbons at a time, okay? So it's kind of a nice symmetry to it. And so this process of burning 2 carbons at a time is called beta oxidation. If you've taken biochemistry in college or medical school or whatnot, you might have heard about beta oxidation. And the reason why it's called beta oxidation is because the enzymes that do the oxidation attack the second carbon in the line, so the beta carbon. This one here is the alpha carbon here, that's the first carbon, and that one here is the beta carbon. So beta oxidation just means the number 2 carbon is getting whacked by the enzymes that do the oxidation process. And so the beta oxidation process attacks that second carbon, and then what happens is each step of beta oxidation releases 1 2-carbon compound, and then what you have here is a fatty acid that's shortened by 2 carbons. Okay. Now, before I talk any further about acylcarnitines, I have to mention acyl-CoA briefly, because I have to explain the logic of measuring acylcarnitines. Now, coenzyme A is a factor inside tissues. It's derived from vitamin B5. It's also called pantothenate, okay? And the business end of CoA is over here on the left. What you see here is a sulfur atom, okay? Now, what happens is when tissues want to break down carbon fuels, okay? You have these carbon fuels, which are like carbon atoms arranged, bonded together in a chain of some kind. What these tissues do is they attach these carbon fuels to the sulfur end of coenzyme A, okay? And you get what's called an acyl coenzyme A. The acyl just means a carbon chain, so that's the carbon fuel the body wants to burn, and it's attached to coenzyme A via this sulfur group. So you form a carbon-sulfur bond. Now, the biochemical reason for this is that the sulfur atom has different electrochemical properties, which weaken the carbon-carbon bonds for the fuel molecule attached to it. Then it makes all the further biochemistry easier for the cell. In practical purposes, this means that any kind of fuel that the tissue wants to burn is going to be attached to coenzyme A, okay? So if you really want to take a look at what fuels tissues are burning, what pathways are active inside a tissue, you have to look at the carbon fuels that are attached to coenzyme A, okay? The acyl-CoAs are the actively burned fuels inside tissues. Now you see here, I have this picture of this bottle. Okay, it's in honor of Anton Chekhov. I have this picture here. So when vitamin B5 pantothenate was first characterized a while ago, I guess maybe 100 years ago, the general feeling that this vitamin in particular was good for fingernail health and for health of your hair. And so some people, clever people, took advantage of that and made this vitamin B5 fortified shampoo, which they call Pantene, right? Pantene for pantothenate. Now, I don't know if Pantene is sold in Singapore, but whenever I go back to the US to visit, I see commercials for Pantene on TV with these models with nice wavy hair, and it just makes me laugh. Okay. So I just said, if you want to interrogate fuel metabolism inside a tissue, you want to measure the acyl-CoAs, okay? Now, the problem, as Jan Hong will tell you, is measuring acyl-CoAs is very difficult, okay? They're unstable, okay? They fall apart very easily, and they're very difficult to measure. Trust me, we tried, and we never have. Bob Stevens was the guy who helped run the Metabolomics lab at Duke, used to tell me, he said, if someone tells you they can measure acyl-CoAs, well, they're lying to you. So very difficult to measure. Now, what happens, and this is another funny aspect of biology, the acyl-CoAs are generally produced in the cytoplasm, for instance, but to be burned as a fuel, they have to enter the mitochondria. And as I teach the graduate students and medical students here at the school, is that the acyl-CoAs cannot directly pass into the mitochondria on their own. The CoA is something about its chemistry can't fit through the transporters to get into the mitochondria. So the cell has devised this kind of clever and a bit cumbersome process where the CoA is swapped out for this chemical called carnitine, which is actually, interestingly, a nitrogen-containing compound. And this acyl-carnitine can then enter through the transporters into the mitochondria, where it rapidly gets reconverted back to the acyl-CoA and can be burned as a fuel. Now, acyl-carnitines are very stable and very easy to measure. So you can take, for instance, a blood spot from a newborn infant and put it on a piece of cardboard and let it dry. So these dried blood spots can then be mailed in the mail to some central lab in the capital city, and then you can extract the acyl-carnitines to look and see if there's an inborn error of metabolism. That's just to illustrate that these acyl-carnitines are very stable and you can handle them and detect them very well. So it turns out it's very easy to measure acyl-carnitines. It's very hard to measure acyl-CoAs. But acyl-CoAs are the thing that the cell's actually burning, but you can't measure it. The general feeling now is that the acyl-carnitines are more or less in equilibrium with the acyl-CoAs, okay? So that when you're measuring acyl-carnitines, you're actually sort of, it's a marker of what's going on with the acyl-CoAs and the fuel metabolism. That seems to hold up quite well. Now, I mentioned these blood spots. So measuring of acyl-carnitines has been employed for a very long time now. It's in the pediatric world for screening neonates for inborn errors in metabolism. And this is one of the seminal papers by Dave Millington, a guy in the US who helped pioneer this method of using the acyl-carnitines to look for neonatal screening of inborn errors of metabolism. And some people thought, well, actually, let me just show you an example. So here's an example. So this is actually a patient of mine from many years ago. And this person would come to my clinic and would tell me about how he get these really bad muscle aches whenever he did heavy exertion. And he was the guy in his 60s, and all his adult life, anytime he'd have to go work hard, he developed severe muscle aches. He had to lie down for a day or two and get better and get back to work. And no one ever knew what was going on. So it seemed kind of suspicious to me. This kind of story of muscle aches after heavy exertion sounded like a metabolic myopathy. So I sent his blood to our diagnostic lab to do an acyl-carnitine profile. And this is what I found. And this is just, we don't know who the patient is, so I can share this to you. So, and I'll discuss acyl-carnitine in just a second, but this is this patient's acyl-carnitine profile. What I show you is sort of what a normal value would be like, and what in white, and in black is this patient's value. Everything seems sort of okay until you get to here. And here you see the C14 acyl-carnitines in this patient are way up, okay? And then if you look downstream, it's way down. So this is very classic for a defect in fatty acid oxidation. In particular, this is the classic pattern for VLCAD deficiency, very long-chain acyl-CoA dehydrogenase. So this person I diagnosed based on this serum acyl-carnitine profile has an enzyme defect in long-chain fatty acid oxidation, which means he can't oxidize fatty acids very well. And so in situations where his tissues need to burn fat, like heavy exercise or prolonged fasting, his tissues are not able to generate the energy necessary because they have a block in the enzyme here. And so the tissue has this energy deficit and tends to die. That's why he would get all these severe muscle aches every time he'd go work on the roof. He was a roofing contractor, believe it or not. So this is an adult example of how you can use acylcarnitines to diagnose disease. Now, of course, this patient had almost complete absence of function of this enzyme. You're not going to see this mostly in your cohorts, but it could be that maybe mild changes in the activity of some of these enzymes don't produce severe disease like muscle aches every time you go out and exercise, but perhaps they do change the function of the tissue in enough ways to cause disease. And that's kind of the rationale for using acylcarnitines to look at human clinical cohorts, to try to identify how metabolism pathways might be changed with different types of human diseases. So this is a kind of pull from an Excel spreadsheet showing the acylcarnitine panel that we will report to you if you happen to want to run your samples with us. And so the last part of this lecture, another 10-15 minutes, is just going to go through this acylcarnitine panel, try to explain to you what all these things mean so you have a better understanding. It doesn't look so daunting, okay? So the first thing you'll see is we say C, right? So C meaning the carnitine. So we're measuring acylcarnitine. So the C represents carnitine. And the number after that represents the number of carbon fuel chains attached to the carnitine. So C2 is a 2 carbon containing carnitine compound. So this is acetate, right? Acetylcarnitine. If you look at C8, that's an 8 carbon containing compound. So that's octanoyl carnitine, I guess, all the way up to C22. Okay, so you have C2 all the way to C22. Let's see. You'll see that for the long chain carnitines, it's all even numbered: C22, C20, C18. As I said before, nature seems to like even numbered stuff. So all these long chain carnitines, which represent intermediates of fat catabolism, metabolism, they're all even numbered. And then you start to get some odd chain ones. See here, C7 and C5 start to appear, and I'll talk about that. There's also these things, colon and then a number. So C8:1, what does that mean? Things like OH, C5OH, or C3-DC. What does that mean? Well, I'll tell you now. Okay, first of all, we measure all these different acylcarnitines. If you get their profile back, you'll see that some are very high abundance and some are very, very, very low abundance. And it's not because our machine gets stuck and we don't know what we're doing. It's because that represents the way that these fuels appear in the body. So this is just something I pulled off of a paper. On the left, it shows you the types of fatty acid chains appearing in a normal human diet, okay? And what you'll see is that the predominant long chain fatty acids that you have in your diet are a 16 carbon, fully saturated, okay, fatty acid. So this happens to be palmitate or palmitic acid. And then an 18 carbon chain fatty acid with partially unsaturated. So this is oleate, and this is linolenic acid, I believe. So C16, C18:1, C18:2 are the primary fatty acids in your diet, okay? And then if you take a look at human fatty acids, the fatty acid content in human adipose tissue, you see the same kind of predominance. So you see palmitate, so C16, fully saturated fatty acid, oleate, so C18:1, so partially unsaturated long chain fatty acid, and C18:2, which should be linolenic, but I can't promise you that. And then the other fatty acids appear but much lower abundance. So when you see acylcarnitine profile, you'll see the C16 peak will be quite high, C18:1 peak will be quite high, and it's because these are the things that are feeding into the pathways, okay? C21 up here, not so much, so much lower abundance. Okay, now, good, I have a few minutes to get on my soapbox, okay? Now, as I explained, tissues normally see a wide range of free fatty acids when they're trying to burn fat as a fuel, and it's going to be a mixture of saturated and unsaturated fatty acids. That's normal physiology, okay? Now, if you do basic science experiments, especially tissue culture experiments, a common experiment people do is they'll expose cells to fatty acids and show what kind of effects these fatty acids have on the cell and make all these reports. A frequent thing people do is they'll throw in a single fatty acid. So for instance, palmitic acid is very popular. Expose cells to palmitic acid for a while and then see what happens, okay? So palmitic acid, if you go back one, is a fully saturated C16 containing 16 carbon containing fatty acid. And turns out that cells don't like it if you just give straight palmitic acid, straight long chain, fully saturated fatty acid. They're not used to it. They're used to dealing with a mixture of different fatty acids. And so some people who are prominent in this field, and this is the paper that they wrote a while back, have suggested that when you do experiments where you're exposing these cells to fatty acids to look at fatty acid effects on cells, that you use a mixture of fatty acids. So this shows table 1. And they suggest a mixture of both saturated and unsaturated fatty acids because that more faithfully replicates real physiology. And it turns out that just adding palmitic acid has a tendency to activate stress kinases and stress pathways, which normally wouldn't happen in a normal cell because there's a mixture of different saturated and unsaturated fatty acids. So that's my soapbox. I'll get off of it now. Now, when I think about acyl carnitines, I try to classify them in different ways. Some people are going to object to it, and that's fine. But when I look at when collaborators come with their data and I look at it, this is what my brain does. So I apologize if my brain doesn't agree with your brain. We can hash it out later. So the way that I look at it is first, I classify it by chain length, okay? So I have arbitrarily said that short chain acyl carnitines are from C2, so 2 carbon containing fuels, up to 6 carbon containing fuels. You could argue that you should stop at C5, but C5, C6, that's about right. Medium chain acyl carnitines go from C8 about to C10 or so. And then the long chain acyl carnitines go from C12 and above, okay? Short, medium, and long, okay? And the reason I do this is because the enzymes that process these fuels have a preference for chain length, okay? So like some enzymes like to do 18 to 20 carbon chains. Some enzymes like to metabolize 12 to 14 chain fuels. Some enzymes like to only metabolize short chains. So there's a biological reason why I separate them out, because if there is a defect in a short chain enzyme or medium chain enzyme or the long chain enzyme, if you separate them out, you can have a better chance of spotting it if it's just a subtle defect, okay? And the other big class are all these things that have OH and DC. I call them the dicarboxylate. The DC here, that's a suffix. The DC suffix stands for dicarboxylate. So these are dicarboxyl fuels, and I'll explain in a bit. And the OH is for hydroxyl, so hydroxylated fuels. And again, I'll explain the meaning for that in a bit. Okay, so if I have a collaborator that gives us a bunch of human samples, serum samples, when we run the acyl carnitine panel, and they let me take a look at the resultant Excel spreadsheet, there's a few things that I do right off the bat, okay? And so I take the Excel spreadsheet and I start to do some sums. So I'll just add up all the acyl carnitines. So I just do on the Excel spreadsheet a function where you add up all these acyl carnitines. So you sum up all the acyl carnitines, okay? Or I do another thing where I just add up all the short chains together. So I do the sum of the short chains, the sum of the medium chain, the sum of all the long chain, the sum of the dicarboxylates. If you want to be fancy, you can separate these dicarboxylates and the hydroxyls into short, medium, and long as well, if you feel up to it. And then you do ratios. So the ratio of short chain to all acyl carnitines, medium to all, long to all. And the rationale, again, is if you have blocks in various pathways, might be more apparent if you look at the ratio of the long chain to the short chain. So if you have a block in the middle, what you should expect is everything longer than that particular enzyme's preference will be elevated, and everything shorter than that particular enzyme preference will be lower. And this kind of simple thing helps you take a look and see if there's differences. And here on the right are these criteria that these geneticists use to diagnose these inborn errors in metabolism, like short chain acyl-CoA dehydrogenase deficiency. They see an increased level of the C4 carnitine, and so they have an actual cutoff value for that. Medium chain, so they're calling medium chain 8, so I guess I'm in the clear for them. Whereas VLCAD, you remember I showed you from my patient with VLCAD, the C14 carnitine is elevated. So everything C14 and above will be higher, and everything 14 and below will be lower in this particular case. Okay, now, I told you about this colon thing, right? So when you have these carnitines, right, I'll go back, you have ones that have like C8 and then C8-1. So what does that mean? That means the saturation of the chain. So you have saturated fatty acids where there's no double bonds at all, okay? And then you have unsaturated fatty acids where you have some double bonds. And then this colon and then the number after it tells you how many double bonds there are in that fatty acid. So here's oleic acid, so from olive oil, the prime constituent of olive oil. And that is an unsaturated fatty acid with a single double bond. So that's called C18-1, okay? This is linoleic acid. It has 2 unsaturated carbon-carbon bonds, so it's a C18-2. And then linolenic has 3, so it's 1, 2, 3, okay? And then to get ahead of myself, some of these ones will be hydroxylated, and so that's where you get this OH. So here is a 18 carbon fatty acid with 1 double bond and then 1 hydroxyl group. So that is C18-1-08. Okay. Now, let me see. Okay, so I'm going to try to wrap it up in the next couple of slides. Ooh, I do have some more slides. Okay, so the dicarboxyl. So the dicarboxyl, what is a dicarboxyl? The dicarboxyl is a fatty acid that has been attacked from both ends. So normally a long chain fatty acid has a carboxyl group at one end and then just a naked carbon at the other end. As I explained to you, the normal beta oxidation pattern, the beta oxidation means you attack the second carbon from the carboxyl group. That's the beta oxidation. There's a whole other kind of oxidation called omega oxidation, okay? In omega oxidation, they are attacking, the enzymes attack the omega, or the last carbon in the tail of the fatty acid molecule, okay? And then when you have omega oxidation, the end result is in that oxidation process is to form a carboxylic acid group on the tail end of the fatty acid molecule. So the end result is you have a normal fatty acid, which has 1 carboxyl group at the head of the fatty acid. When you start the omega oxidation process, you add a carboxyl group to the tail of it. So now you have a dicarboxyl fatty acid. You have a carboxyl at the head and the tail. So this is a dicarboxyl. So these dicarboxylated fuels are a product of omega oxidation. So if you see changes in the appearance of the dicarboxyl carnitines, these carnitines with the suffix DC, that means you have changes in omega oxidation, okay? Now, it turns out that these dicarboxyl carnitines pop up a lot when we do these studies in humans, so large human cohorts. This is just one example, I think a good prominent example. This is work done by Swati Shah's group at Duke. And they had this study called the cath-gen study. So every patient that came into the Duke cath lab would get some blood stored for doing DNA and metabolomics work. And then they would follow these patients prospectively. They did metabolomics on all these blood samples, and the way Swati Shah likes to do her analysis is use principal component analysis. That's one way to do this data analysis for these metabolomics data sets. And her analysis showed that there's one principal component, one factor from principal component analysis, which predicted recurrent cardiovascular events. So the people that had elevated levels of the metabolites in that one factor had much higher probability of having recurrent cardiovascular events, okay? And if you look at that factor 8, so this is the factor 8, it's a dicarboxyl carnitines. So these patients with established cardiovascular disease that went in for some testing, the ones that had higher levels of these dicarboxyl carnitines had much higher risk of recurrent cardiovascular events, okay? So this is an example of where these kind of obscure things, this product of omega oxidation, is popping up in human disease. And it turns out that we see a lot of these dicarboxyl signals in our human cohorts. I think because this dicarboxyl, this omega oxidation, is a fundamental alteration that happens in a lot of diseases that are related to metabolic syndrome type problems, and suggests that changes in omega oxidation and fatty acid oxidation in particular are driving some of the pathogenesis of cardiovascular disease. Okay, now the OH hydroxyl group is generated when you're trying to deal with fuels that have branched chain. So these are called branched chain fatty acids. You see here, there's an extra carbon sticking out, and that is a problem for the usual oxidation enzymes that deal with fatty acids. And so there's a different kind of oxidation called alpha oxidation, where you attack the alpha carbon, the first carbon in the long chain fatty acid, and the intermediate is the formation of a hydroxyl group. So increased accumulation of hydroxylated species is a sign of increased alpha oxidation of fatty acids. Now, interestingly, alpha oxidation occurs primarily in the peroxisomes. Peroxisome is an interesting subcellular organelle, which people don't think about too much, but it's kind of the junkyard where you have to deal with a lot of these inconvenient types of fuel and molecules. So there are diseases associated with peroxisomes. I think the one that is most well known is probably X-linked adrenoleukodystrophy. This is a real disease that we see in the clinic, and these affects boys. It's X-linked, primarily affects boys, and they develop progressive neurologic decline, and then adrenal dysfunction as well. And it was a basis for this movie called Lorenzo's Oil. So this is a movie from the 1980s. For those of you who you know, are a bit older, starred Nick Nolte and Susan Sarandon, and it was a real life story, quite an interesting story. It was an Italian couple whose boy was diagnosed with X-linked adrenoleukodystrophy. And they weren't biochemists; they were just sort of lay people, but they looked into the disease very closely, and they found that one of the problems with this disease is that you get overproduction of very long-chain fatty acids, and that's harmful to the person. And they discovered that if you give oleic acid to these people, you block the sort of competitively inhibit the activity of these elongases that produce the VL, very long-chain fatty acids. So essentially, giving oleic acid, so giving olive oil, is able to slow the progression of the disease and somewhat improve the symptoms. And so that's why they call it Lorenzo's oil, because the boy's name was Lorenzo. And so basically olive oil, giving him Lorenzo's oil, olive oil was able to help cure his disease. So interesting bit of 1980s pop culture, a movie from the 1980s. It's kind of interesting because, you know, I teach medical students and grad students, and they're a lot younger than I am. And I've tried to talk about pop culture in my classes before. And what I've discovered is my students have never heard of John Wayne and never heard of Ferris Bueller. So it seems like the younger generation, for them, history began with the invention of the iPhone. Before the iPhone, it was just this vicious, savage, dark age, not worth considering. Just my old grumpy self. Okay, a few more things. I guess we're almost ready to wrap it up. The odd chain fatty acids. If you look at the acyl-carnitine profile, we have things like C3, C5, and C7. So they're odd chain, okay, because they're odd. I told you mammalian biology likes even chain stuff. So where are these coming from? So a lot of this comes from the gut bacteria. So fermentation of gut contents can produce odd chain fatty acids. If you're a cow, for instance, a ruminant, you have a lot more of these than a human, but we don't do much of that, so we don't have as much. Some are plant-derived, and the other source is breakdown of BCAA, which I will address now. So the C3 and the C5 carnitine is an important one to look at when you're looking at these panels, because it is a signal of amino acid catabolism. And the reason why is that when you break down the branched chain amino acids, leucine, isoleucine, and valine, that pathway is well described. Two other things that you produce during the breakdown of the BCAA are propionyl-CoA. Propyl is a 3-carbon compound. So propionyl-CoA is a 3-carbon compound, okay? And its cognate acyl-carnitine is a 3-carbon carnitine. So the C3 carnitine is primarily being generated by the breakdown of isoleucine, which generates propionyl-CoA. Similarly, when you break down valine, okay, the end result is succinyl-CoA. So succinyl-CoA is part of the TCA cycle. It's a 5-carbon containing fuel. And so the C5 carnitine is a succinyl carnitine. It's primarily generated from succinyl-CoA from valine. So when C3 and C5 carnitine are increased, it's a pretty good indicator that what you're seeing is increased catabolism of amino acids. And I mentioned this story back at the start of the talk, way back when, the BCAAs and metabolic disease. This is that same table from before. I was showing you the data showing that people who are obese and insulin resistant have elevated levels of valine, leucine, and isoleucine in their blood compared to the lean, insulin-sensitive control. What I didn't show you then, what I'm showing you now, is they also have higher levels of propionyl-CoA, propionyl carnitine, and the C5 carnitine. C3 and C5 carnitine are also elevated. So they have higher levels of the BCAA, and they also have higher levels of the carnitines, which are derived from BCAA catabolism, okay? But you can see this for other things. So there's different kinds of fuel switching that go on in the body, but higher C3 and C5 suggests catabolism of amino acids. Okay, very close to being done. C4OH, so C4OH is a 4-carbon with a hydroxyl group. So this is primarily from beta-hydroxybutyrate. Beta-hydroxybutyrate has 1, 2, 3, 4 carbons and this hydroxyl group. And this is a ketone, okay? So generated from the process of ketogenesis. So C4OH is an indicator of ketogenesis, okay? And I mentioned before that ketogenesis is becoming kind of interesting because things like the SGLT2 inhibitors cause a ketogenesis. SGLT2 inhibitors cause increases of the C4OH carnitine, so you can track that in your human studies. I was trying to think about ketones in human disease, and this kind of was sort of attention getting. This table on the left is a large meta-analysis of studies looking at the use of ketogenic diet to treat children with intractable epilepsy. These are kids who have bad epilepsy. Their seizures are not controlled on multiple anti-seizure medications. And it turned out that putting them on a ketogenic diet was very successful in controlling their seizures. And really no one understands why. I mean, people know the brain can use ketones as a metabolic fuel, but why these particular kids can have their seizures controlled on a ketogenic diet is unknown. But I think very effective, very simple intervention, very effective. And as an interesting side note, I have here on the right the typical ketogenic diet for some of these patients. If you could look, breakfast is whipping cream, eggs, bacon, fat, and heavy cream, right? Lunch is heavy cream, mayonnaise, ham, cheese, right? So that's a ketogenic diet to treat these kids. Okay, now one or two more things. Acylcarnitines, looking at disease models. So I'm an endocrinologist. I'm interested in diabetes. And sort of one of the great, I think, really unanswered questions in diabetes is why this happens. So this is a graph showing BMI, body mass index, and risk of developing type 2 diabetes, okay? And you see right when you hit to BMI of 30, so this is considered the cutoff for obesity, you see a massive increase in the risk of developing diabetes. So why is it that as you get fatter, you become more insulin resistant? Why is it when you become fatter, you have this higher risk of developing diabetes? Well, to be honest, we don't know. I mean, people have been banging away at this for a very long time, and I don't think there's a very good explanation. But people have tried to give explanations and have produced models. And one of the currently favored models for how insulin resistance works was born out of some studies using acylcarnitines. This is work done from Debbie Muir's lab at Duke. And she was looking at these animals fed a high-fat diet who become insulin resistant. And if you look at the acylcarnitine pattern in these animals, regular diet is a black bar, and the white bar is a high-fat diet. You see large accumulations of long-chain acylcarnitines in these high-fat fed insulin-resistant animals, okay? And so what this is indicating is that in the tissues of these animals, they're burning a lot more fatty acid fuel. There's a higher reliance on fatty acid fuel. And so through a number of different experiments, which I won't go through, they built this model suggesting that with increased obesity, you get this increased pressure of beta oxidation, which kind of messes up the mitochondria and causes mitochondrial overload and energy overload, mitochondrial stress, which then drives the insulin resistance signal in the cell. So this is one of the models that people favor for insulin obesity-associated insulin resistance. It's not the only model, but it's one of the ones that was brought out with the use of these acylcarnitine panels. And I think this might be the last data slide. So I'm stealing a bit of JJ's thunder. So JJ will be talking next week about data analysis. JJ and Lim Suchi work at Kutek Plot Hospital, and they have this large cohort of people with diabetic kidney disease. He did a very comprehensive acylcarnitine analysis of these patients with diabetic kidney disease, and he found a lot of interesting effects. And for instance, the people who have diabetic kidney disease have a high accumulation of all these dicarboxyl carnitines, for instance, okay? And there's some other effects with the long-chain acylcarnitines, the short-chain acylcarnitines, as well as some other things that we can measure that we're not talking about today. But as you can see, with diabetic kidney disease, you see accumulation of these dicarboxyl carnitines, so they're raising their head again, suggesting changes in fatty acid oxidation pathways, changes in omega oxidation pathways, maybe changes in endoplasmic reticulum, which are potentially contributing to the pathogenesis of this disease. Okay, so this is my last actual slide. So where is this field going to? I think if you're a person who wants to do this kind of work in the future, you should look to do this kind of thing, which is combining metabolomics and genomics. There's very few papers out there so far. I'll keep an eye on it. This is one of the first ones, again, done by the group in Duke, led by Swati Shah, okay? And this is follow-up for the data I had showed you earlier, where when she looked at people who have high risk of recurrent cardiovascular events, their acylcarnitine profile showed accumulation of these dicarboxyl acylcarnitines, okay? Now, that's at the same time that they were taking blood for metabolomics, they took blood and did genomic analysis, okay? And then what she did, which was very clever, is she looked at the level of these dicarboxyl carnitine in each of these patients, and then they looked at the DNA, and the question they asked is, are there SNPs in the genome which control the level of these dicarboxyl carnitines, which we see accumulating in these people with high levels of high risk of heart events? And they were able to find a couple of SNPs which seem to help control the level of these, I don't know, not pathogenic, but these metabolites which are dangerous, which accumulate with heart disease. And so they call this metabolic quantitative trait loci. So instead of looking at heart attack as the trait that you look for SNPs for, use the level of these metabolites, which are themselves then connected to the disease. So you have an intermediate between the gene and the actual disease, which is the metabolite levels, which you can measure and then interrogate your genomic DNA to see if there are SNPs that can control the level of these metabolites, and perhaps then this will allow you to connect genes and gene areas to the actual disease. So this is, I think, one of the first applications I've seen for this. If you're someone who's collecting human cohorts and you happen to have genomic information and you have metabolomics information, I think this is sort of a natural way forward, and hopefully we'll see a lot more of this. I haven't seen too many publications yet. I'm wondering whether it's because it doesn't work, or it doesn't work very often, or people just haven't cottoned on to it yet. So I guess time will tell. So now I'm asked to give a plug for next week. Our friend and colleague JJ, Janjun Yu from Kutekpot Hospital will give a talk about how to do data cleanup and data analysis when you have these human clinical cohorts had metabolomics done.
Okay, thank you, JP. Thanks everyone for staying. We are a little bit overrun. Please feel free to stay for Q&A, or if not, please feel free to also email us the questions. We'll get back to you. You can email to either JP or me directly. Would anybody like to ask questions? From Shigeki. Hi, Shigeki. Number one, do you expect BCAA supplementation to have positive effects on muscle activity in healthy individuals? Obese or IR subjects have higher BCAA levels due to reduced catabolism by muscle.
So that question actually isn't really worked out. Actually, Tan Hong Chen from SGH has done some stable isotope tracing studies, which did suggest decreased catabolism. I'm not sure about the answer. I know someone else here, Kuching Ming at NUH, have been feeding people a diet with elevated BCAA. And when he measured the BCAA levels in the blood, there was no difference. So to actually cause changes in BCAA levels in the blood by feeding more BCAA, I'm not sure that's going to work. I think because you'd have to force feed them so much BCAA to change the level in the blood that it wouldn't be practical. In terms of normal healthy people, I've always said if you want to look at nutritional intervention, you should look at these bodybuilders and weight builders, because they take this thing to the extreme. I mean, they do things that we wouldn't be allowed to do by our IRB committees. And if you look online in that community, one thing they do is they always have massively, they take supplements that are massively supplemented with BCAA, because they have this feeling, and it may be correct, that if you're doing bodybuilding and want to increase muscle mass, you need to have lots of BCAA present, because that's one of the major storage forms of nitrogen which is put into your muscle. So a healthy person who's working out and all that and taking horse pills and supplements, yeah, BCAA probably works. For the rest of us, I don't think it makes that much of a difference. I mean, I walk to the MRT every day. I do my 8,000 to 10,000 steps. But for me, BCAA supplementation, I don't think it's going to do much difference.
And the second question is, I think for me, is it more challenging to measure C25 and C22.6 in the acylcarnitine form? I don't think it's too much of a problem. With my skills, yes, I'm able to do it. Just joking. I have to try it out, but I think it's okay. Yeah.
Those are very low abundant species.
Yeah.
I mean, they're like 1/10,000th the level of C16 and C18, for instance. But I mean, if it's part of the biology that you're interested in, yes.
Mm-hmm. All right, any more questions? If not, we can, oh, one more from Ivy, a naive biochemistry question. Oh, no questions, naive. Is nitrogen cycle highly influenced by circadian rhythm?
I don't know. I don't know. I mean, I think Chan Hong, a long time ago, you worked on fatty acids, right? With what's his name? And are affected by
Joshua Goodley?
Yeah, right. Fatty acids were affected by circadian rhythm. I mean, you would expect it to be because your recycle is going to be active when you're taking in protein, for instance. So it should be higher, during mealtime as opposed to during fasting. But when you're fasting, you might need to harvest amino acid for gluconeogenesis. So I guess, I don't know. Wake me up in the middle of the night and we'll take some blood and measure.
All right. One question. From Shuchen, we'd like to clarify in one of the slides, how does choline relate to nitrogen metabolites and CVD?
Well, choline is one of the components of like these red meat diets. Choline is one of the things you find in the red meat. Interestingly, carnitine is also highly abundant in red meat. So carnitine, choline, a lot of these phosphocholine, these are sort of methylated nitrogen compounds, which the gut bacteria work upon. So it could be choline. It's probably other nitrogen-containing compounds, which the gut bacteria can then convert to TMA and then liver to TMAO. And that's the link. Interestingly, because I worked in a group that was, before this, that was looking at acylcarnitines, and we thought maybe we could supplement free carnitine, give people extra carnitine to help smooth out the metabolic sort of traffic jam that's happening in the mitochondria. And it actually worked. In mice, it worked very nicely. In humans, it worked okay. But then all this study with nitrogen in a diet causing heart attacks makes me have second thoughts about giving people lots of carnitine. Because what if they convert it all to TMAO and then die of heart attacks? That would feel really bad. So it gets very complicated when you start adding in all these different layers. I hope I answered that question. Maybe I didn't.
All right. Okay, I think we'll stop here. If you have any further questions, please feel free to email us directly. Thanks again. I hope to see you guys next Friday, where JJ will talk to you about using statistics to analyze your humongous set of data. See you next week. Thank you.